Our focus today is going to be to brush up on a cell and the organelles inside of the cell. This is important in anatomy because we know that everything living is made up of cells, and those cells make up the tissues, and tissues make up organs, and organ systems and organisms, and that's what we're studying are the larger structures, but we can't neglect looking at the basic unit of structure and function for life, which is the cell. So we know that the cell is made up of many organelles, and those organelles have different functions and different structures inside of the cell. So let's take a look at what those are, just to review. The nucleus is the largest structure generally within an animal cell, and it has a chromatin inside of it, and the chromatin is what gets wound up to make chromosomes, and inside the chromatin are segments of genes that are segments of DNA that code for particular proteins, and that's what makes you who you are and came from your parents. The nucleus is surrounded by a nuclear envelope, and it holds the nucleus, but it also contains pores, and these pores allow large molecules such as messenger RNAs to go out into the cytoplasm. The nucleus itself contains a nucleolus, and some cells have more than one nucleoli inside of them, and this is the site of RNA production, and RNA is the ribonucleic acid that's responsible for helping to make protein. Transport is done by the endoplasmic reticulum, and it stretches from the nuclear envelope to the cell membrane, so it's a passageway between those two. Its job is to transport proteins. If it is considered rough, it has ribosomes along it, and if it's smooth, it does not have ribosomes. We know that ribosomes are the site of protein synthesis. The Golgi body looks like a flattened stack of pancakes, and this is where the proteins get packaged and ready for export, and the vesicles are formed from the Golgi. The cytoplasm. Cytoplasm is gelatinous substance that holds the organelles in place, and we used to think that it was just a gel-like substance. We now know that it actually is made up of a cytoskeleton and helps keep the structure and the form within the cell. And the cytoskeleton is composed of microtubules and microfilaments. The mighty mitochondria are the energy producers inside of the cell. They are double membraned, and they make ATP. They're known as the powerhouse, and they have inner folds called cristae, and the substance inside of the mitochondria is referred to as the matrix, and this is where ATP is produced. Lysosomes, their job is the cell eater and the cell caretaker, so if there's any bacteria and viruses, these packets of enzymes are going to open up and kill that virus and take it in, or the bacteria and take it in. It also, when it's time for apoptosis and the cell dies, it's going to open up and rupture, and the enzymes will break down the components of the cell into reusable parts or smaller parts that can be disposed of. And your white blood cells are just packed with lysosomes so that they can engulf bacteria that come in or viruses that come in and dispose of them. Peroxisomes aren't one that we talk about often. They also have enzymes inside of them, and it helps the cells to digest things. Notice they have a double membrane around them, a lipid bilayer that's a double membrane layer, similar to your cell membrane. Centrioles are found in animal cells only, and their job is to form spindle fibers, and spindle fibers are what allow for mitosis to occur and the chromosomes to be pulled apart inside the cell. Cilia and flagella are found on cells, not on all cells, but on a lot of cells. The cilia, there's many short bundles, and the flagella, there's normally only one or two, and they're both hair-like, and their function is movement of the cell. Vacuoles contain waste and water, and inside of a plant cell, it helps to maintain the structure. Inside of an animal cell, we may have small vacuoles, but we don't really look for the large one like we do, because we don't have to worry about in an animal cell carrying out photosynthesis that requires that large amount of water. The cell membrane and the plasma membrane are one and the same, and the whole purpose is to keep the cell contained. It's selectively permeable, which means that size is going to limit, and not only size but also the polarity of a molecule limits what can go into and out of the cell. It's made of a phospholipid bilayer, and the two things, like I said, that limit how permeable it is are going to be mainly the size and the shape. And this is a nice picture of a plasma membrane, and you'll notice that it is a double lipid bilayer. And we see the hydrophobic tails on the insides, hydrophilic or water-loving heads on the outsides. We see the cholesterol that keeps the fatty acid tails from sticking together. We see proteins that are throughout it, and this is the way that it communicates with the outside, the receptors that are out here, and this helps us to get things into and out of the cell. And these are the microtubules inside the cytoplasm.